everything is in Canvas. I have all the stuff for Lab 1 for you guys under the Lab 1 for today's date. This is an old document. I need to just delete it. But I actually have a copy of the textbook, right? I have the same thing for Lab 2. By Lab 2, you should have your textbook, though, right? But for some reason, you don't get it till like the day before lab, and you want to be a responsible student and start working on your homework right after this class, you have access to this. As well as on reserve at the library. And then again, the Science Resource Center, I'll make sure they have an up-to-date copy, so if you're in there working with friends or you're over at the library, you can check it out from the reserve desk. You can sit down there and work on your stuff. It's a really great place. If you haven't been there, definitely go check it out. So any handouts that I give you, I'll also post them on Canvas in case you lose them, so you'll see that one that you have in front of you is already here. The lectures I usually don't post until after lab. The first couple of labs, while we're getting into the swing of things, I will make them available to you. But then after that, unless you're a student with disabilities, you're not going to have it until after lab, right? Because it's going to have the answers to the homework in it, right? I'm not going to give you all the answers before class. But, that's a great guide. And then as you see right now, I'm recording it. As I said before, if you miss lab, you watch the recording, then we make up the lab, right? So it helps save time. So I already downloaded the PowerPoint, so I'm going to open that up. So we're going to talk about first what's covered in page 6 of the current lab manual, which unfortunately has a typo in it, but my scanned copy online has the correction as well as the one in front of you right now, so you don't have to worry about fixing that just yet. It's not necessarily a formula you need to memorize, but as you guys will learn from me, I try to present the information in multiple different formats so that we meet all learning styles as best as possible, right? So some people, equations make sense. Other people's cute little diagrams or drawings or examples help, right? So I try to do a variety of explanations for the different things we need to know. So we're going to talk about the value of controls because they're very important. And this is discussed on page 6 of your lab manual. The newer edition that we're going to be going to in the fall, they finally fixed this typo. I've only been telling them for like 2 editions. I don't know why. And then we're going to do Ubiquity of microorganisms together as a class. I'm going to go relatively fast, but as I said, you already have access to this. You can see that, right? So you can go home, you can fill this in as you're studying for next week's quiz, right? Take your time. I even have a practice quiz to help you learn the terms, right, in Canvas. So you can do over and over again. It's not for points.
When you were just saying that the textbook is available on reserve, do you mean the lab manual or the textbook that goes along with the class?
We call it the textbook. It is a lab manual. Okay. Either term can be used synonymously, but it's only one book. Okay. It is technically a textbook as well as a lab manual. It is all that you need for this course. Where some other courses, right, you have to have a textbook and a lab manual. That's where people get confused. For us, it's just one thing. Okay. Has everything you need, which is great. Okay. And then we're going to introduce the light microscope. One of the things I'm going to ask you to do for homework, because we're going to actually take out and use our microscopes next week, is to watch a video that I've done on how to use a microscope. Because how many of you guys have focused all the way to oil immersion before? Exactly. 1 person, 2 people, right? So this is a new technique. It can be difficult, especially if you're not comfortable using a microscope. So it'll help you and help all of us if you watch this before class. So I'll show you that link. So a couple terms. And again, don't fret about definitions. You will find them sometimes in the text or in the glossary of your lab manual. You can look them up online. The important thing is that you write down something you understand, right? I'm not going to have you memorizing definitions verbatim, right? And again, I'm going to give you the definitions in my PowerPoints, right, to help guide you. But if you need to reword that for your studying, do so. Again, I'm not going to collect your homeworks. I'm just going to look at them, okay? Those of you guys that like to type, you have to print them yourself if you type up homeworks, right? Because you have to have the paper in lab. You can't bring your laptop in, your iPad, or anything like that, because again, you're not going to want those types of things in this room, okay? But you can do that. I have one student, I always have a student every semester that doesn't like their handwriting and can't read their own handwriting, so they type up all their notes. That's fine, right? But you have to print it and bring it to class because you need it in class. So what does false positive mean to any of you guys?
Positive
Right, it's not right. False is definitely not correct, right? So this is where you expected a negative result, but in fact it came out positive. So the example that I use for all of these terms, real world example that help us kind of grab onto this, is pregnancy tests. So guys, don't tune out on me, right? You're going to like some of the interesting information I found out about pregnancy tests. So this is where, right, you take the test, right? You're pretty certain you're not pregnant, but it comes out positive. How the heck is that possible? Well, it has to do with these two other terms here, known as specificity and sensitivity. Certain tests have a certain range of sensitivity, right? How much stuff you have to have for it to come out positive within that test. And what actually has to be there. How specific is this test, right, for what we're testing for? So false negative, of course, is the flip of that, right? Where it comes out negative and you were expecting what? A positive result. Can this happen with the pregnancy test? Absolutely, right? So that deals with, when you have false negatives, that deals with sensitivity. So this is the limit of detection of that test. So for the pregnancy test, what are we actually testing for? Anyone know? Human chorionic growth hormone, which is typically only produced when the fetus is implanting into the uterus, and that's a hormone that's used to develop the placenta. So under normal conditions, right, that's going to be produced. It's actually in your bloodstream. But what filters your blood? Your kidneys, and produces urine, right? And that's why you can take a urine sample, because the excess stuff is going to end up in your urine. Excess, right? So we have to get to that level that there's enough in your urine that it's detectable by this test, right? And this test really, they've really amped up the sensitivity. I remember you had to wait like, I don't know, a couple weeks after your first missed period before you could do a home pregnancy test and it'd be accurate. Nowadays, I think you can do it as soon as a week after or 5 days after your first missed period. So really small quantities, it's sensitive. It can really detect. And you don't end up with what's called a false negative, right? Could you be pregnant and it come out negative? Absolutely, because there's not enough for it to detect, right? So then you go to the doctor's office, and anyone know what they're going to test instead of your urine? Your blood, because you're going to have higher levels earlier on in pregnancy, and that's going to be more accurate. Okay, can we have a specificity issue? This is where we end up with a positive result, right? And we were expecting negative. So guys, if you pee in the cup because you feel bad for your girlfriend or wife or whoever, right, and she's got to do the pregnancy test, oh, I'll pee in the cup too. Or a different cup, I hope. Should it come out positive? No, last I checked, guys don't have babies, right? Not quite possible just yet. They'll probably figure out a way to do it. This movie's about it, but it doesn't really happen. But guess what? One day, a guy, I guess, felt bad for her, peed in the cup, and it came out positive. Oh, crap. So, of course, the doctor's like, how did I do that? Right? This is supposed to test for human chorionic growth hormone, and it's coming out positive for a guy who shouldn't produce this hormone. Turns out, apparently, there's some product that you produce when you have testicular cancer that will show up positive on the pregnancy test. So they're actually using it in doctors' offices now as an early indicator for testicular cancer. Who knew? Right? I don't know exactly what compound you guys are producing that's similar enough that this test is detecting it. But it is doing it. Quite interesting. So that makes sense to you guys, right? And so we pretty much know, right, or we find out in the case of the pregnancy test, what our sensitivity ranges are, right? How good is it detecting at small quantities of what we're looking for, and how specific it is. For the most part in this lab, we deal a lot with sensitivity, right? We don't get enough of the product produced to get the result of positive that we're looking for. There's one instance where we have a specificity issue, where if you don't you don't perform the procedure correctly, then you don't know what's causing the positive result, right? But that's a procedural problem that we can get around the specificity issue, right? And so we'll talk more about that when we get to that actual test. It's the catalase test. So those are controls. Sometimes we need to go one step further. We need to have standards. Controls, typically we have positive and negative controls, right? The pregnancy test, does it have a control? Yeah, if you actually look at the strip, because now they have those little digital readouts, but if you remove it and you look at the strip, if you're pregnant, how many lines are there? 2. 1 for the positive control, 1 for your test sample, you're pregnant. I was so excited I took a picture. But not always the case, right? If there's just 1 line, it means the positive control worked, the test worked properly. No other line, you're negative. Within the confines of the sensitivity of that test, you're potentially negative, right? It is a pretty specific test. You don't need to do like Juno did and take like 6 or 8 of them in that movie, right? If it comes out positive, it's probably positive. Go figure out what you're going to do about it, right? It's a pretty specific test. That's yes or no, right? And if there's no lines, it means the positive control didn't work. The thing is going to read error, right? Repeat test. There's something went wrong. They can't give you a solid yes or no, positive or negative, because the test didn't work properly, right? And that's really one of the most important things about controls, other than for you to see what a positive and negative looks like, which is a lot of times why we do a lot of the controls here, because you really need to be able to discern one from the other, but also to make sure that the test is done properly, right? Especially multiple step tests. That make sense to you guys? Sometimes we want to go one step further. We want to know how much hormone you've produced, or how many antibodies you produced against the vaccine you've received. So some of you guys going to nursing school nowadays, they don't just want vaccination records, they want titers. They want to know how many antibodies you have against the things you should have been vaccinated against. And we're having a problem with one of the vaccines right now. Anyone know what's spiking? And yet we have a vaccine against it. What disease that most of us have probably never heard of? Starts with an M. Mumps. We're having a problem with mumps right now. And they say that the vaccine is, I just forget what the CD says. CD said, I think 70% effective. So there's an issue there, right? And I've had some nursing students that have had the vaccine 3 times, never produce antibodies. So it's a multiple facet problem. People hadn't got vaccinated, didn't get properly vaccinated, didn't get properly boosted, and then you have that group of people that it just doesn't work for. So especially if you don't get vaccinated, those people aren't protected, the ones that can't get vaccinated or it doesn't work for. And if the rest of us don't get vaccinated, then we have all these people that can be susceptible and pass it on to others. Right? So it's a serious problem. So how do we get a number? Well, we have to have standards in that case. So kind of like a control, except now we're going to have a number associated with it, right? How much protein, how much antibody. And typically, again, you're going to do a dilution of your positive controls, different concentrations. And you're still going to usually do a negative control, especially for these types of tests. There's one test that we do later on in microbiology lab. Yeah, clearly. It's okay. Have a seat. Don't sit there. Sit at the lab bench. So you have the materials you need. It's fine. We'll get you caught up. Okay, so a lot of times we use a machine, and there's a test later on in the semester. I want to convert that into where it's a quantity, a number, instead of just is it positive or negative. So, although I'm fearful it won't happen this semester. But we will use this machine throughout the semester called a spectrophotometer. It actually can read the levels of color and give you a number for it. So especially when you have a color change, the machine will actually give us a number, and that number equates to how much it is there. So if we have our standard, and of course, you can graph this out. The good news nowadays, no one does this by hand, right? Computers do it. You plug in your numbers, right? It creates the graph. You get your number for your unknown sample. It compares it to the graph on the known data, your standard, right? And you can extrapolate out about how much was in the sample. So for this example of a protein that read 0.45 on the absorbance reading on the spectrophotometer, is around 8 milligrams of protein that was in that particular sample. So that's how they figure out your titers, right, too, is that they have a standard control for the number of antibodies, and then when they test your particular sample, that's how they're able to get a number, right? It's pretty cool stuff. So, remember I said I'll put things in different ways. So in your book, they even have these cute little pictures. So if you observe a positive result, and you expected a positive result, you're happy, right? Because it was a true positive, right? You expected positive, you got positive, your positive control works. Well, what happens if it comes out positive and you were expecting a negative result? That's a false positive, right? And that's an issue, and it's usually an issue of what? Specificity. Think back of the guy getting the positive pregnancy test. How the hell did that happen? Right? Something else is causing it to be positive. Right? We have a problem. If you have a negative result, you see a negative result, and you were expecting a positive result, again, this is a problem, right? This is a false negative. This is your positive control, and it didn't come out positive. What happened? You probably have a sensitivity issue. You don't have enough of that substance for it to register positive in that range of that test. Now, of course, if it comes out negative and you were expecting negative, you're happy. That's a true negative. It did what it was supposed to do. So remember, this is the formulas that's messed up in your book. It makes sense that if all you have is true positives, right, and this is 0, you're going to have 100% accuracy for your test in the sensitivity range. Because remember, false negatives are an issue of sensitivity. Is any test 100% sensitive? No, but we know the ranges, and it's because people have done out the experiment numerous times under numerous conditions. They've calculated out the sensitivity of a particular test, especially ones that are pretty well known. So we want zeros here, but we don't always get them. We don't always not get false positives either, which again is going to affect our specificity, right? And again, not all tests are 100% specific. Look at what happened with the pregnancy test, right? Somehow, some hormone, some substance a guy produces when they have testicular cancer came up positive on a pregnancy test. And it's supposed to be testing for human chorionic growth hormone. So it happens. And for the most part, we either know why these things happen or how they could happen, or of course in that case we're going to investigate what happened. Anyone know what ubiquity means? Everywhere. So this is ubiquity of microorganisms, and we pretty much know this already. Where can you find a microorganism? Everywhere. They're everywhere. They're in the air. We're going to demonstrate that today. They're on your skin, in your body, on the desktop, on that pen you're using, definitely on your cell phones. Right? They're everywhere. And we're going to demonstrate that today because recognizing where they are, which you're pretty much usually already aware of, will help you understand why the techniques we employ in this lab are so important so that we don't contaminate our culture. As I mentioned earlier, we don't work with pathogens anymore because what do they do? If someone says something's pathogenic, what does it cause? Illness, disease, right? So we want to avoid that. But notice these are ones that we know cause disease. Which brings us to our next definition, non-pathogenic. So that means that they don't cause disease. We don't know that they cause disease, right? Has there been organisms that people have worked with and all of a sudden they link it to a particular disease? Absolutely. Happens more often than not. So again, although we're not working with known pathogens in this lab, and we are pretty much working, for the most part, with organisms that are pretty well studied. We also take precautions so that these organisms don't change and become pathogenic. You could bring a pathogen in and not realize it. So again, we're going to practice proper aseptic technique as if we were working with pathogens, right, so that we're as safe as possible. Opportunistic pathogen. This one's interesting. They need what to be pathogenic, to cause disease? An opportunity. The most common ones that take advantage of an opportunity are ones that we refer in an informal grouping known as enteric bacteria. Anyone know where those are found in your body? Enteric. What does that sound like? Intestines, right? Entrails is actually where they get the name from, right? So, guys, don't phase out on me. This is the most guys I've had in the lab. It's pretty cool. I like all people of all types, by the way. I'm not prejudiced in any way, or at least if I am, I try not to be. All humans are prejudiced whether they want to believe it or not, right? It's just whether you choose to control it or not is another issue, right? Okay, so don't tune me out, guys, because I'm going to talk about girls, but you may be a dad or help take care of a girl someday, and this is a very important girl issue. All right, ladies, what are we taught when we're younger about poop? You must wipe in what direction? Front to back, right? You don't want to go forward. Why? What two holes get you in trouble? Well, the anus is where the poop comes from, right? And you don't want the poop to get into the vagina or the urethra, right? We have two holes. Guys just have the one, right? As far as pee and semen go, right? And usually there's a pretty large distance between the poop for guys and the whole peeing semen thing, right? So unless you're really messing around with the poop, it should not get in that other hole, right? Girls, on the other hand, very close proximity, y'all, we got this serious problem. So we have to learn this very serious procedure when we're very young, okay? If you're not taught it properly and you screw this up and you get feces in your vagina or your urethra, it can spread into your body where it does not belong. Because actually your intestinal tract is just a tube through your body. It's actually considered an external environment. If it leaves the intestinal tract, is when it really can cause some serious problems. So although most of these guys aren't going to create a problem, right, and poop can go out and come right back in, believe it or not. I had a student of mine that didn't pay attention to his kid one day, and he ate his own poop, and he's like, why didn't he get sick? I'm like, because he just put it back where it came from. And animals will do this to get normal flora from each other, right, and establish their gut flora. And gut flora are really important to you, right, because they help protect you from infectious disease. Because they're taking up the food supply in the spot, and the bad guys can't get in. Now back to girls. There was a little girl in Louisiana several years back, died as something as that simple. The bacteria got into her urethra, spread to her kidneys. The infection was so bad that by the time they took her to the doctors or the hospital, the doctors couldn't save her. Right? She died. Right? And they linked it back to her own gut flora. And the only way it would have got there is if you didn't wipe properly. So it is a serious issue, right? A lot of times we don't like to talk about poop and all that cool stuff, right? But it is important stuff. Okay, if you're a host, everyone's over at your house, right? What does it mean in terms of microbiology, though? It means you're housing the microorganisms, right? You're the host, right? Any organism that serves as a habitat for another organism, such as a parasite or a commusal. Most of us understand what's the problem with parasites. Why don't we like them? What do they do to us? They suck us dry, yeah. They eat us, right? And in such a way that they harm us, right? That's a parasitic relationship is a harmful relationship, especially for the host. Not necessarily so much for the parasite, right? They're benefiting. My parasitologist friend refers to my son as my ectoparasite, right? He was once an endoparasite, now he's an ectoparasite. I don't feel that way. I feel that the relationship falls into a different category. I don't consider it parasitic. Commusal, I believe, doesn't truly exist. Although they say that that relationship we have with our normal flora is commusal. Which, to help some people remember, communism, right, is based on that some type of similar interaction between two entities. And what is the belief there? Is that everybody gets the same, right? Nobody benefits, nobody's harmed. I don't believe that it can exist, right? I can't think of any true examples in nature where you're not benefiting and you're not being harmed, right? But a lot of times, a lot of texts say, that synergistic relationship. But in truth, when you look at our normal flora, especially our gut flora, they're benefiting, right? They're getting food and a place to live. And we're benefiting not directly but indirectly by their presence. Because they're there, parasites, right, are less likely to take up residency because they have to compete with the good guys that are already there for that parking spot, for that food, right? Mutualistic, I think of a different word. Anyone know what word starts with M that talks about this type of relationship? That's mutual. A good marriage, right? Both partners should what? Benefit from the relationship, right? This is where both benefit from the relationship. Reservoir can be kind of tricky. What do we think of when we think of reservoir? Water, right? Storage of something, right? In this case, we're talking about storage of what? Right, microorganisms. And usually what we're concerned about is the ones that cause disease, right? So any non-human host, typically, or site in nature that serves as a perpetual source for pathogens. These are things that we usually want to avoid. Could a water source be this? Absolutely, there could be pathogens there. Could soil be pathogenic? Yeah, these are things in nature. Can animals harbor pathogens that they could pass to you? Absolutely, right? What is the one virus we were terrified of that they could pass to us? Bird flu? What about most animals, even your companion animals? Rabies, right? And in Louisiana, who do you stay away from as far as rabies go, other than companion animals? Bats? There's one more that's actually top on the list. Raccoons. They're cute. Leave them alone. They're out during the day. They're not supposed to be. They're nocturnal, right? If they're coming up to you, they're not supposed to do that. Stay away. I'll tell you, at the vet school when I worked there, every raccoon they brought in, they killed it, even the babies, right, and tested it, and 90% of them had rabies. Don't mess with raccoons. Or bats. They're bad news. Okay. Another term that sometimes you'll hear is vectors. So these are other things like fleas, ticks, right? Mosquitoes that can transfer disease between organisms. And if it's free living, it does not rely on a particular host, right? No other living organism. Because of that, most of those we don't have to worry about, right? Because if it doesn't need a host, it's probably not going to do what to us? It's not going to make us sick. It doesn't need us, right? It doesn't need a host. Okay, so we've been sitting for a while, right? So we're ready to take a break. And I need 2 volunteers. I need 2 people to go outside and get some samples for us, for our lab. I need 1 person to rub the backside of a leaf with a sterile swab, and another person to put it in the soil. Who would like to do that? Okay. Who's going with you? So go get those samples and we come right back in. Okay. Who wants to stick it up their nose? Wait, we're doing a leaf in what? A leaf and in the soil. Dirt. Nice and moist stuff. Should be good out there. You want to spray? Okay. So really rub it up in the nose. Yeah, really up the nose. Okay. Your mouth along your gums. Yes, go make yourself. We'll make you have to do it now. I'll do it all the time. Alright. Be loud. You get there.
Take the restroom.
Alright. I'll rub it along your gums, yeah. I'll let you rub it on the plate and go to the bathroom. So then gently rub this all along the surface of the plate. You can put that in the regular trash on your way out. Just rub it on the surface of that. Just rub it all over the surface. That's good. Okay. Go ahead and throw that in the trash on your way out. Any other time, most of the time when we're working with stuff, you'll have to wash your hands, but today you get it right. Alright, so we haven't had enough volunteers. Are we good? Alright, you ladies. Rub all over the desk. so we gotta do fingers the whole class, and I need one person, just one person that's gonna touch the plate. Anyone wanna do that? So just press your hand right on the plate. Not too hard. You'll break the arm. That's okay. That's good. That's yours. Hold on to it. The rest of us are going to each touch this. Don't touch this too hard, but you'll see it does have a little bit of give to it. If you push on it too hard, you're going to break the surface. Okay. 2 of you. So grab that. And what you're going to do is just take that swab, right? Those of you guys that have swabs, use some petri dishes. Go ahead and rub, right, on the surface. Go ahead and sit at your lap bench. Rub all over on the surface of the agar, right, not on the lid, right, with your swab. And then the swab can go in the regular trash, that's fine. Hold on to the, okay, and there's one plate that's going around everybody's touching. Oh, did someone set a timer for me for 30 minutes? Raise your hand if you're doing it for me. Okay. Let me know once you started. Okay, once you're done with your plates, go ahead and, as I said, put your swabs in the trash. Cover back up your lid, and then you're going to tape it shut. If you did if you did the nose or mouth, you're going to write your name on it. After we tape these shut, you flip them over so that you write on the bottom. You never write on the lid. The lids, as you can see, can be separated. Did you start my timer yet? Oh, yeah. Okay. Ready? Go. Okay. So this one we're leaving open for 30 minutes to the air. These two I'm not opening on purpose, and I'm taping shut. Anyone know why? These two I didn't know. Control. What kind of control, though? Because if I don't open it, what's not going to happen? What reaction do you decide to have on this list? Something to grow, right? Because guess what's in here? Food. Right? So we're going to do lots of food, and so that'll grow in very large numbers, so you have to be able to see what's in here. Okay? So, there's Sharpies on each table. What we do is we write it along the edge of the bottom of the plate, right? So that we can easily see what may be in the center of the plate. So in addition to maybe your name, right, what might be something else we might need to write on a plate?
Wherever the sample came from.
Yeah, whatever the sample came from, right? So in your case, it is your name along the edge and mouth. So I wrote negative control on these, right, so we expect no growth. What's another important part of information I think we need to write on everything we make in the lab? Yeah, someone's going to come up with it. And what is today again? 18.
Can we write on the tape?
Yeah, we're going to tape it shut, but go ahead and write on it for now. Okay. So write mouth and the date. Okay, so I have negative control, the date. Are we the only class that uses this lab? No, so in case our stuff gets mixed up with someone else's, we're going to do W, and you can write on the tape, 8-8, that indicates, because I don't do section 1, 2, 3. I do day and time, right? So Wednesday at 8 o'clock. Okay? Yes, and so you're either leaf or soil, depending on what it is, right? We're writing what it actually is, right? Wednesday 8, we're writing the date. There's one other bit of important information we're going to write. So we have 3 incubators for use in this room. This one is the most popular. It's 30 degrees Celsius. Anyone know what that equates to as it relates to us? Body temperature. Right, so this is nice and warm, right? This is obviously less than that, and I actually don't have it turned on. The heating element cracked on this one. This one used to be about 30. And I usually put it on to see what the temperature actually is. Sometimes it's above 25. But basically, this is a room temperature storage cabinet, right? So we don't need a heating element. These don't have cooling elements, right? So we just have that one off. This one, on the other hand, I will turn on once we start using, and hopefully it will read 30 degrees. This is a little bit warmer than room temperature, of course, but cooler than body temperature. It's more like skin temperature. We have a few organisms that prefer this little bit warmer temperature that we utilize this incubator for. So these are the incubators we use in the lab. The only temperatures we're going to use today is room temperature and body temperature, 37 and 25. So the unopened plates, I have two of them because we're going to use we're going to do two different temperatures, right? So we need a control for each one of the temperatures, yes? Maybe? I hope so? Okay. So one's going to be 25, and one's going to be 37. And I'm going to put them in our 37 and 25 piles. Alright, so we did two desktop ones because we're probably going to do two different temperatures. Y'all are waking up. So decide who's going to do 25 and who's going to do 37. Make sure that we don't do the same temperature for both. And write it on the bottom of your plate. And then go ahead and put it in the 37 or 25 pile. Okay. Yeah, take a shot. Can stuff that grows at 37 grow at 25? Yeah. But why do we sometimes put them at that warmer temperature? Why do we do that? Why would we take something from our body and not just grow it at nice, cool room temperature? Why would we put it at 37? Yeah, that's the temperature it prefers, right? And it'll probably grow best. Some of them will still grow at 25, but not as well, right? Temperatures we know affects things. We manipulate temperature all the time. We manipulate, that part is not going to come out good. We manipulate temperature all the time in our houses, right, as it relates to microorganisms. You don't even realize that that's why you're doing it, right? We use refrigeration to slow them down. We use freezing to stop them, but I get news for you, they start right back up. You don't kill them by freezing them, unlike us. If you freeze me, I'll die. And we do what else with our food other than make it cold? We cook it, right? And certain foods we cook to certain temperatures, right? I don't usually can remember this, so my thermometer for the meat has all the different ones on it, right? Because for a particular meat, there are particular pathogens, and we know there's particular degree ranges that we need to reach, right? So for poultry, we've done this recently, so I remember, right? It needs to be 165 degrees Fahrenheit, right? So my husband stuck it into the chicken that was in the oven. It came out 170. We're good. Take it out. We can eat it. Internal temperature. This has been worked out for the known pathogens. If we get too hot, what can we actually do to them with heat? We kill them. We can actually kill them with heat. We can actually kill them with heat. And that's why we use the Bunsen burner, right? We're going to incinerate and kill them, even endospores, with using flame, direct flame. Right? And that's why we use it in the lab. So if we're doing the sample of the air, what temperature are we going to put it at? Room temperature. Okay. Why wouldn't we put it at 37? Yeah, but could stuff from the air, from our bodies, end up in the air? Absolutely. The question is, what do we want to see, right? So both answers are correct, right? You could put it at 25, you could put it at 37, right? My last class, I did it at 25, so actually I'm going to change it up for this class, and I'm going to do 37. So we can do a comparison between the two different temperatures, since I have two classes, right? Normally, I do that one at 25, right? But I'll tell you, I didn't get really any growth on the one from yesterday, so I'm kind of curious, you know, what might be in the air that grows at 37 better, right? Will we see something? Hopefully not too much, right? We're hoping the air is relatively clean. Okay, soil. Same thing, right? Really depends on what we want to see, right? In this case, we're going to go with 25. So if you have the soil sample, write 25, put it in the 25 pile. The back of the leaf, what do you guys think? Yeah, 25. Although, I'm pretty sure it's warmer than that most times outside, right? Than about 70-something degrees. Okay, the one person that pressed their hand onto the plate, what temperature do you think we would want to put that at? What are we interested in finding out? Yeah, we could do room temperature, but we're actually going to do warmer because we want to know what's on your hands, right, that may get inside of you that could make you sick, right? So we really want to, like, and that's also why we tape these shut, because we don't know what's going to grow, and we're not going to open these back up and expose ourselves to them. It also keeps them from, getting contaminated when we tape them shut. So that person is going to do 37. Where's the one everybody was touching? Does everyone touch that plate? Where did it end up? Where's our touching plate? Where'd the touching plate go? Nobody knows where the touching plate went? They have it? Okay. Everybody just gently touch it. That one, since all of us are touching it, it's going to have a lot of microorganisms on it, right? So do we want to put it at 37? We could, but it's going to have a lot, right? So we can keep it from overgrowing by putting it at a cooler temperature. So we're going to do room temperature for that one. When you guys are done, I'll mark up that one. Or if someone doesn't mind writing class fingers on that one for me. For the single person, you wrote your name and fingers, right? So we know how dirty you are, right? Next week, just kidding. All right. So nostrils and mouth, though, these are definitely internal things. So what temperature do you think? We're going to do 37. So make sure you mark 37. Make sure you put it in the 37 pile. So go ahead and finish those up, labeling them right. So we have name if we need it. We have source. We have date. We have class as W8 for Wednesday at 8 o'clock. And we should have what temperature, and make sure you put it in the correct pile at the back of the room on the paper so that I put them in the correct incubator. Okay. One thing I want to point out is that most of the media we're using is nutrient agar. This is a pretty general purpose media. Just about anything will grow on it. Except for the person who did their mouth, we are using a blood agar plate because I want to show you guys ahead of time. And hopefully I remember to come in here tomorrow and put it in the refrigerator because the reaction doesn't really stay long term for next week if I don't refrigerate it. And I actually have my plates from yesterday's class, and I'm pretty excited about some of those results there that I may pass around to you guys in a moment. That contains whole red blood cells, that blood agar plate from sheep. And the microorganisms you have in your mouth that everybody has, even if you just went to the dentist for a cleaning, have the ability to produce enzymes that will damage red blood cells. And this is particularly the reason why some of you guys, if you have heart defects or certain medical conditions, when you go to the dentist, you may have to take antibiotics before you go, because inevitably, right, you always end up bleeding. They poke you somewhere somehow. It always freaking happens, right? And then those organisms from your gums can enter from your mouth into your bloodstream, and they can cause serious problems. Because some of them, and everybody has this one in your mouth called Streptococcus mutans, which is the one that contributes to the decay of your teeth as well, because it converts sugar into acid, which eats away at the enamel of your teeth. That one will poke little holes in the red blood cells, and that plate's going to turn green. Others will completely annihilate the red blood cells that we'll see later in the semester, and all the red is gone in the plate. You can actually see through it. But the green one's kind of cool, and so I wanted you guys to see that, you know. And again, if I can link it to a real-world situation, then you remember it more, right? And later on in the semester, when I show you green plates again, guess what? I swab my mouth for you guys to see the green. Because right now we don't have, but I did order Streptococcus mutans. So, but it was just a cheap, easy way in the past, right? I just used my whole mouth for you guys. But there could be other stuff there, right? It's not a pure culture. So Why did I not open those plates? 2 of those plates, I didn't open them. What are they for our experiment? There are control, and more specifically, there are... What type of control? Negative control. That's important. They show sterility of the media, that this food that we started with was sterile, which means devoid of any microorganisms. No growth should happen on those plates, right? They should come out negative. And this is what I'm excited about, y'all. Both my negative controls from yesterday have no growth on them. So that means our media to start out with was not contaminated for yesterday's experiment. So when we're asking for an appropriate label, we're talking specifically about a negative control or no exposure in this case, to be a little bit more specific. What are other labels that we need to put on things that we produce in lab? What's important information that we put on these plates? What do we put? Date. Keep going. What type of sample? What class it is? What temperature, right? Degrees that we're going to incubate it at. And again, as I demonstrated and as we've done, we write it along the edge of the plate so we can see what grows on the center of the plate. Always on the bottom of the plate, never on the lid, right? Because the lid could be separated to the bottom, especially if we don't tape it. So next week when we come back, or I'll know tomorrow, if growth appears on both those unopened plates, what does that tell us? The sterile media wasn't sterile, it was contaminated, right? So either the person that made it or the media they used wasn't in fact sterile, they used proper technique. Any number of possibilities could happen. How does this affect our results? So if you were in a research or clinical lab and your negative controls ended up with growth on it, can you proceed? No. You have to start over, don't you? Crap! Because none of your results are reliable, right? Because you don't know if that growth from his fingers was from the contaminated media or his fingers, right? We can't separate that out. Controls help validate our experiments, right? And if they don't come out as expected, then we can't trust those results. Well, we're doing 2 different temperatures, so we have the 2 different temperature controls, right? So what if, say, one of them has growth and the other does not? Can we trust our results? No. And again, are organisms affected by growth? Temperature. Is there a growth affected by temperature? Sure. And so some of them will or will not grow at different temperatures. So it might be because, whatever our contaminant is only grows best at 37, so only grew on that plate. Does that mean it's not in the 25-degree plate? No, it could be there, right? And again, where the contamination happened, right, you can extrapolate out and potentially find out. In these classes, we usually talk about that, right? Because sometimes we will get negative controls that are positive, that have growth on them. We won't repeat it, right? We're not doing a true experiment. We're just trying to show to you guys the different types of growth that can happen and where sources of contamination are for your cultures, right? Which, as I said, most of us nowadays understand microorganisms are everywhere. And I have a really good example later on in the semester of a picture I took of a student I know she breathed on her plate because it was a blood agar plate, and it has beta hemolysis in one tiny little spot, and the rest of the plate is her organism that she was working with. And Staphylococcus aureus, which grows in some people's noses, is beta hemolytic. So she probably breathed on her plate. What we did today, I let you guys just completely open those plates, but from now on what you would do is hover that lid over your plate, or even I hold my breath for a second so I'm not breathing on my plate, so the microorganisms from my breath, from my nose or my mouth, do not land on my plate and contaminate my cultures. So these are some of the growths we've seen in the past. And as you can see, you do have a variation in growth. And again, why are we picking all these sources? We want to demonstrate that microorganisms are found just about everywhere, right? And these are sources of contamination for our culture that we need to look out for. What type of organism is this? It's not bacteria, is it? It's fungus, it's mold, right? And for the most part, and this is a lot of times why we do the outside samples, because we do have a HEPA filter we run in this lab, and we try to keep mold spores out of here so they don't contaminate our cultures, because they'll really make a mess. It's also why we tape these plates shut, because if we do get any spores, we're not opening them. If we are, we're doing it very carefully, because we don't want to release them into the environment and contaminate our lab. So we'll see different patterns based on the different temperatures, right, and sources. And you can definitely see, look at this one was 25 degrees of the air, and this was 25 degrees of the desktop. Notice the things that are falling out of there on our desktop, right? And then as opposed to on our desktop at 37, things that probably come from our hands look similar, right? Similar type organisms. So why were we asked to incubate at 2 different temperatures? Right? Because we're looking at those 2 different temperatures. We want to know, and again, we use Celsius scale. This is the conversion. You don't need to know it. You need to know in general, right, that 37 degrees Celsius is pretty much body temperature. 30 degrees is about 86 soil skin temperature, right? So our soil microbes that we use in this class, we put them at 30. And then room temperature, right, around about 77 is about 25. Sometimes it's higher than that in here. So where are the ones that grow best at 37 probably coming from? From us, yeah, from our body. Can they survive at 25 degrees on our desktop? Yeah. Microorganisms are really good at starving, much better than we are. Obviously not a lot of nutrients on our desktop, but when we take them and put them on those petri dishes with a lot of nutrients, they'll really grow like crazy, especially when you put them at that preferred temperature. But some of them are really good at starving and actually go dormant. Anyone know what those are called? Some bacteria can form this special structure. Endospores. Or some scientists like myself are kind of bad and just say spores. But those guys really are 100% resting, and then they reactivate once they enter into a host and realize that there's food available. Some of these cause really scary diseases, right? That we want to avoid. One of them we actually get vaccinated against every 10 years. Anyone know? This one's a spore former found in the soil. What disease does it cause? Tetanus. Clostridium tetani. Clostridium difficile, another spore former in the environment, will cause the infection known as C. diff in the intestines. Really nasty, smelly infection. I thankfully never have smelt that. I hope I never do. I avoid hospitals like the plague. If you see me in a hospital, I really give a shit about that person. Because I won't go there. What's another one? And it could be in your food. Nope, not a spore former. Botulism. Clostridium botuli. Oh, and then the one we worry about for bioterrorism. Starts with an A. Anthrax, spore former. We've done a really good job in the United States of cleaning up cases of anthrax, so the spores aren't really found in the United States.
So when disinfectants have the recommendation of like letting it sit for 10 minutes on the counter, is that to kill endospores?
No, you're not using any steriliants or high-level disinfectants at home, hopefully. You haven't stolen any from any hospitals or bought them where you should not buy them from. You don't need that level of clean, and for the most part, any endospores that get into your house that might be tetanus would be the only one you have to worry about, and that would be if you have a wound that goes anaerobic, and hopefully you've kept up with your vaccination status, so then you don't have anything to worry about. Good question. Botulism, on the other hand, can be a problem, and you can solve that problem if you cook the foods that you home can, right, to the correct temperatures. Because you can deactivate the toxin that it forms. You're not going to kill the endospore, right, but you're going to deactivate the toxin that causes the problems. So why do we get different appearing colonies on these different plates? Because they were grown at different temperatures. Temperature definitely does affect what type of organisms you're going to select for the growth of. So each organism has its own temperature profile, its cardinal temperatures. It has a minimum. It has an optimum, its very favorite best, and then it has its maximum, and this is what we take advantage of, right, with cooking. If we exceed maximum, we can actually kill microorganisms. Not only impede growth, but we can terminate them. all microorganisms are actually put into 5 different classifications of preferred growth temperatures. So ones that fall into these ranges have optimum growth temperatures that fall into these particular ranges. Anyone know which one we would be? Yeah, so we prefer 37 degrees Celsius. So what would that be? Mesophiles. We're right there in the middle. Right? We're Goldilocks. We want it just right. Right? Some microorganisms, especially ones that can grow in your refrigerator, create problems like listeria. Right? Our psychotrophs and even psychrophiles are the really crazy ones. Psycho actually means cold. I think that if you like cold, you are crazy too. Right? Psycho is usually what we consider crazy. And then we have the ones that are at the really high temperatures, right? You don't need to know these temperature ranges for this class. I go over this more in my lecture class. But just to make you aware that we actually have organisms can live in these really different temperature type environments. And we have to take these into consideration when we're dealing with those types of organisms and what we're trying to achieve. So why don't we make things sterile? So anything that we use in lab, we're going to put in a rack labeled trash. I'm going to put in the 25-degree incubator. Our lab prep person that helps out with that will actually take it over to one of the biology labs. If you've been in biology room 108E, you've probably experienced our stink that we share with them and the heat from the autoclave. I'm so sorry, but that's where they were set up. So the machine looks something like this. You may have seen it in the back of that room, or if you walk by, you could look and see we have 2 or 3 machines back there. They're relatively small. So one, are we going to even be able to fit the desktop in there? No. Do we need these desks to be that level of clean? No, we don't need it to be sterile. But any of the organisms that we produce, right, we need to kill them before we dispose of them, right, because we don't want to contaminate the environment. And so all the cultures that we work with are going to be properly disposed of by our lab prep person. And then also we're going to use this to sterilize our media so we don't have contamination, right, of the cultures that we're trying to grow in this lab. We're just going to use Lysol. It's a very good disinfectant for our lab benches. And as I said, those paper towels can go in the regular trash unless we have a microbial spill, right? Unless we spill a large number of microorganisms, then those paper towels have to go in our spill bucket. Okay, I'm not going to go over all the parts of the microscope. I'm definitely going to ask that you guys go over these on your own, right? You have access to this. Watch the video on how to use a microscope. We'll go through the steps and actually use the microscope and look at lots of slides next time. Magnification, these are like some made-up numbers they have in your lab manual. It's a very simple formula for figuring out total magnification. It's your ocular lens, which is the one you look through, times your objective lens, which is the ones you can change. So these are really crazy numbers. These are the actual numbers for our microscopes. So our ocular lenses are 10x. This is fixed, right? These are the ones you look through. And then we have the choice between these 4 different lenses. The 4x scanning lens, because you can see a large surface area, but not very magnified. It's only 4 times by the 10, only gives you 40 times magnification. Really is a waste of time in microbiology lab to look through for the most part after next week. But the nice thing is it's a really tiny lens, and it gives you a huge amount of space between your stage that holds your slide and the lenses. So you can very easily take your slides on and off at scanning and not have to keep focusing over and over and over again, right? Once you have your microscope focused once, you just switch out slides at scanning. And you put your microscope away at scanning with your stage all the way down. We typically will start out at low, which is the yellow ring, which is, again, only 100 times magnified. For microorganisms, this isn't a whole lot when we get to bacteria. Thanks to me, and I found our cleaning kit, all our high dries you can actually see through. I cleaned them all, right? Or I replaced them with spares that I had. So you can, if you want to, actually look through the high dry and see something. High dry means you do not put what on it that I had to clean off of all of these? Oil. The next one, which we will live and use all the time, don't ask me after next week if you need oil. The answer is yes. The answer to oil is what? Yes, because you're always going to be looking under the oil immersion lens. That's the highest magnification. That's where you have the greatest possibility of actually seeing bacteria, which is what we work in primarily in this lab, right? So we have pre-cut lens paper so you don't use too much, right? And the oil up front, and we will teach you how to properly use that in this class. This is 1,000 times magnified, but which, believe it or not, is going to still feel really freaking small. Really small. When you're done for the day is when you clean off that lens. If you goof, right, and you get oil on the high dry lens, just wipe it off right away so you don't forget. The reason why I say that too is why you don't forget, because at the end, if you don't screw up and you follow it properly, you have no reason to wipe that lens. Because what some students sometimes do is they wipe the oil immersion first, which is definitely going to have oil on it, right, if you're actually seeing what you're supposed to see. Then they use that same lens paper and they wipe oil onto the high dry. So again, if you're cleaning up, you got to go high dry first and then oil. But if you don't get it on the high dry, you don't have to clean it, right? Ever have to clean it. Just the oil when you're done for the day. You don't clean in between each slide and go through a whole stack of lens paper. When you're done for the day, you clean it and you put it away. Okay, so some important calculations, they're actually not that hard, right? Relate to how our microscopes work and how these lenses work. So numerical aperture is actually a number. It's going to be listed as a decimal. So this is the condenser. Anyone know what that lens is and where it's found on your microscope? The condenser lens. It's under your specimen in the stage, and it focuses the light from the light source, which is a light bulb for our microscopes, onto your slide. And then helps focus it into these objective lenses. So it's down here. It has a numerical aperture. All lenses do, especially ones you're using a view. So for these guys, you'll see the X for the magnification, and then next to it, that decimal number, is its numerical aperture. The best way to understand this one is it's basically the prescription. Oh, it's the timer. Close the lid. That's allowed to go off. Just close the lid to that one that's open. And if you don't mind labeling it for me, I'd appreciate that. And remember, we're going to put that one at 37. We're going to deviate. And it's 30-minute air exposure. Thank you. So, I need glass person. Do you need those to see me? Okay, take them off, please. How many fingers am I holding up? Oh, you're pretty good. Okay, go ahead and put them back on. He's right. Usually, sometimes I won't get this. They were blurry, though, right? You actually used more than your eyes to see. You probably calculated it was too thick for one finger, right? That's what he did. He didn't actually see that it was two. Why? They're too close together. And his natural lenses in his eyes cannot adjust on their own. Either they've gotten that way over time, like it happens to us as we age, right? I should actually wear reading glasses, but I just blow everything up onto computer screens, so I don't have to wear glasses, right? But his lens, our lenses are flexible, but his can't flex for distance, and so he wears additional lenses, corrective lenses, to be able to see clearly distance. Can these lenses in our microscope change shape? No, they're fixed. So they have a fixed number, just like corrective lenses have a prescription, right? And sometimes that has to change because the lens in your eye changes, and again, you've got to correct for that. So does that make sense to you guys? This is telling you how good that lens can focus. And it's a number value, like a prescription for sunglasses, or reading glasses, or prescription glasses. With this number, we can calculate what's called resolving power or resolution. Limit of resolution or resolving power. This tells us resolution. How good is that lens at helping us discern two objects that are very close together, like my two fingers? This number you get for resolving power or resolution is a distance. A minimum distance that must exist between those 2 points for you to see them as two separate images. So of course, if I spread my fingers like this without his glasses on, he'd say two. Right, he'd see two blurs. Because they're a pretty large distance apart. That part makes sense to you guys? Because this is very important. This is a minimum distance. That asks us a couple of questions. I'm going to actually skip ahead in my PowerPoint because I find if we do the calculation and then we talk about these different situations, it makes sense for most of us. My last class agreed, so I'm going to skip ahead after this slide. So this is our formula that you do need to know. It's not too complicated. You guys can memorize this. I know you can. It is the wavelength of light measured in nanometers, and this is where we get our distance. Because you notice there's no units for numerical aperture. It's just a decimal number. There are two things that determine the light for your specimen. The focusing of that light, the condenser, and whatever objective lens you're using. So those two numerical apertures have to be added together at the bottom of the fraction and the wavelength of light you're utilizing on the top. And I said fraction, didn't I? It's not that bad, y'all. Trust me. Typically, we use blue light filters. It's not going to make things blue, but it's going to focus most of the light within that spectrum of the light, visible light spectrum. And the reason for that is because it's a smaller wavelength. This will make more sense as we do our calculations. Because small in this case is good, right? Small is good. We want small distances when it relates to resolution. Okay, so I'm going to skip over this one and this one. And let's do a calculation. So they give you all the information you need. They give you the wavelength and they give you the two numerical apertures. So we just set up our equation and we calculate it out. I do have calculators for those of you guys who are mathematically challenged that are used in the lab only. So again, you don't use your phones, you don't contaminate your phones, right? And things like that. And you don't bring your own in. So if we've set this up correctly, we end up with 347 nanometers for the resolution under these conditions. That means I can put two objects how far apart and see them? How far apart do they have to be for me to see two objects as two separate and not blur together? 347 minimum, right? Could I go higher than that? Sure, because then they're further apart, right? And we're going to resolve them. This is minimum distance. So if, as the example says, if they're only 330 millimeters apart, are you going to resolve them as two separate images, or are they going to blur into one? What's our minimum number? 347. What is this? 330. Is that more or less than 347? It is, in fact, less. So they're going to what? Blur. Right? They're not the minimum distance. Everyone still with me? Okay. Yeah, this is why, corrective lenses have a particular prescription, because they got to adjust for these things. Otherwise, you don't see things as separate and they blur. Okay. So, another example, under these conditions, which I think is high dry maybe, 248 nanometers. So that's our minimum, right? That's the number that everything's going to be at or above for us to see. Nothing less than that. So if things are 250, is that greater than 248? Yes. So they're far enough apart. It's close, right? But they're far enough apart that we would discern them as two separate images. For our microscopes, the resolution is 200 nanometers, or 0.2 micrometers. This is about the size of a bacteria. Hence why we've got to do oil immersion, right? The human eye can apparently go to 100 micrometers, right? This is not nanometers. And apparently if you have really good eyesight, you can visualize 10 lines between a millimeter line on a millimeter ruler. I'm not that good. I'm lucky I can see the millimeters. We will measure in millimeters, right? In this class, we do have rulers, and we will be doing that type of measurement. So again, we can't see this stuff with our eyes, not in the detail that we want to see them as individual bacterial cells. So we've got to employ oil immersion. Now back as it applies to why some of these things matter. So we want a small wavelength of light. We want to use blue filters. Because our fraction, remember, What part of that fraction is the wavelength? The top of the bottom. The numerator, the top, right? So in this case, right, we want a small number. We actually want the small piece of pi, right? Which one's the smallest? 1/6, because the top is the smallest. Can we change the bottom of this equation, really, for our microscopes? That's numerical apertures, right? Can we change the lenses? We can change between lenses, but that lens itself, can we change it? No, we're stuck, right? So, depending on how good your lenses are, but also another problem we run into is light itself. Because we have to stay, for light microscopes, within that range of visible light. Because if it's not visible, are we going to see it? No. So we can't use UV light, right? Because you can't see it, believe it or not. You see things that are affected by UV light, like fluorescence, where the light excites the electrons and produces fluorescent color. And we have special microscopes that utilize fluorescent light, UV light, to see fluorescence under those conditions. Electron microscopes, are you seeing anything? No, a computer is generating it. And then we've got really fancy microscopes now that I haven't had a chance to play with. They use atomic force, where you can go to the atomic level for molecules. Pretty crazy stuff, huh? Does that make sense to you guys? Right? We got to stay within that range. And again, typically we use blue filters, right? Because that's the smaller of the wavelengths. But it's not going to make things blue. But if you look at the bottom of your microscope, there's a little blue filter down there, right? To give us that wavelength. Okay. So as it relates to microscopes, we use what's called bright field microscopy. The background is bright. Usually the organisms are stained or have color to them. They absorb a lot of the light, and we're able to visualize them. We don't employ dark field microscopy. This is where the background is, of course, dark, and the organisms are illuminated. I would like for us to get phase contrast. We use special filters. So there's bright, there's dark field, here's phase contrast. Do you notice all these little things outside the euglena? That's because the filters are affecting the light waves as they're traveling, and we can see subtle differences much easier with that type of microscopy. We don't currently have that here, but hopefully soon. And then fluorescence, I think they might have across the hall. I need to check with them and do a tour one day. They don't have. I don't know what Zoe can do.
He's using pros, but I don't know if they have.
Yeah. Well, that's for the PCR. But there's some organisms that are pretty cool that fluoresce. Pseudomonas aeruginosa, although you can find it in the soil outside, we don't mess with it in the lab because it's a pretty serious pathogen. But it was fluorescent. So, what do you need to do for next time? Finish filling out Lab 1, right? Study it for your quiz. I'll tell you I won't be that heavy on terms, but to help you start practicing, because guess what you have to do in this class as it relates to terms? You have to know how to spell them and the organisms, so I have lots of in-Canvas practice quizzes. Quiz 0 is the one to help you with the terms, learn the terms. I'm not going to ask you a lot of terms. I'm actually more concerned about the questions that we talked about, right? And how to calculate numerical aperture, how to interpret resolution, how to calculate resolution and interpret resolution. How do you do magnification? That one's pretty easy. The two objectives times each other. How important controls are. Yep. You need to complete the second one, right? So go ahead and take a look at the second one. So notice we're going to have a lot of terms. Again, you can look on the internet, you can look in your book. As long as it makes sense to you, you write something, it's fine. Notice the first one I got from the internet, that site isn't working anymore, but I like that definition. If you get it from some other source, you might want to write where you got it from. Because if it's really wonky, I might ask you that, like, where the heck did you get that from? Wikipedia, remember, isn't usually necessarily a good place, although I'll tell you, most of the microbiology stuff is pretty accurate. You're going to read through that exercise and fill in the blanks for the outline. And then the last 3 pages, it tells you to draw and label from the pictures in your book. Guys, do I collect this? No, I'm going to look at it, make sure you did it. So you have some idea what you're looking at, and you don't have to have your lab manual out, right? And you have some idea. I'm going to have nice, pretty color printouts, and I might even put my answer key online for you guys ahead of time. This is for you. This is an important part of the learning process, right? Some of us, we actually learn by drawing stuff out. I'm not going to judge you on your drawings. If you're one of those people that like look forward to this and use colored pencils, like, great. I don't care, right? This is for you, but do it, okay? Don't not do it. Don't be like, oh, I'm just going to go buy pictures and I'm just going to pull pictures off the internet. Don't do that. Draw, okay? Because then I know you actively looked at it and thought about what you're looking at. Does that make sense to you guys? Okay, watch the microscope video, right? Because we're going to actually use the microscopes to look at these things under the microscope. All right, and then these are here. These recordings will be here too to help you guys.